Thanks everyone for joining us, and I'll make a few announcements as we get settled. So I wanted to let everyone know that we recently released an RFA for a new PHIND seed grant opportunity. So we hope that many of you will be interested in applying for that. We look forward to that. So the eligibility is anyone that has PI status, and the scope is essentially anything within the precision health domain. So we have some of the details here. We've sent an email out, and you can also reach out to me should you have any questions. So we look forward to seeing what comes in for that. Just to give you a future upcoming symposium. Next year on March 23rd, if you save the date for PHIND Symposium. And here we have a number of exciting speakers to give us updates on PHIND projects, as well as some new faces you'll see to talk about their work in precision health. And we also have some great upcoming PHIND seminar talks looking at the microbiome in regards to Alzheimer's disease progression, as well as looking at infant epigenetic markers. So we're looking forward to those as well, and hope you'll be able to join us. So without further ado, I'll hand it over to our PHIND director and chair of radiology, Dr. Gary Gold.
Thanks so much, Ryan, and welcome everyone to the October Precision Health and Integrated Diagnostics Seminar. Today, we're very fortunate to have Andrew Lipchik giving our seminar. Andrew majored in chemistry at Xavier University, where he performed research on the development of oxygen activation nickel complexes with Dr. Craig Davis and Dr. Michael Baldwin. He went on to obtain his PhD from Purdue University under the mentorship of Dr. Lori Parker. His thesis work focused on identifying determinants of kinase substrate specificity. This understanding was applied to the development of novel kinase-specific peptide biosensors to monitor intracellular kinase activity. Andrew joined us at Stanford following his graduate work, where he joined the laboratory of Michael Snyder in the Department of Genetics. His focus here has been on understanding the impact of the immune system on insulin resistance and glucose metabolism. In today's seminar, Andrew will speak to us about IgG glycosylation as a scalable biomarker of insulin resistance and metabolic health. Andrew, thank you so much for speaking with us today.
Great. Thanks for having me. What I'm going to talk today about is the mitochondrial function as it is related to IgG glycosylation, and how this can be leveraged as a scalable sensor for monitoring insulin resistance. So to begin, the problem with type 2 diabetes and insulin resistance starts with diet. And I think it's best summarized by this photo of 45,000 pounds of sugar in Times Square, which represents how much sugar is consumed by children in the United States every 5 minutes. In addition to that, the epidemic of insulin resistance and type 2 diabetes continues to outpace projections globally, where we have already outpaced the projections made 20 years ago as to the incidence of type 2 diabetes that would be reached by 2025. Diabetes is not only a problem of scale based on the incidence, but also the cost. If you consider the combined revenue of the major sports teams or leagues in the United States, their revenue last year was $31 billion. compare that to the $760 billion that was spent on diabetes worldwide, that's 24.5 times as much, and it's projected to increase to 845 billion by 2045. This cost also extends to life expectancy and incidence or causes of death, where diabetes is among associated with the 4th leading cause of death worldwide, but insulin resistance is also associated with a number of diseases, including cardiovascular disease, cancer. It's newly being investigated as having a role in dementia, as well as liver disease and kidney disease. So taking together this association, insulin resistance could be responsible up to 35 million deaths annually. So due to this impact on overall health and contributions to disease and the cost, it's imperative for prevention and early intervention and detection of insulin resistance. So there have been a number of trials that have aimed at trying to prevent and delay the onset of type 2 diabetes when patients have impaired glucose tolerance. And these studies have examined the use of alpha glucosidase inhibitors, which prevent glucose absorption, the use of metformin, which decreases hepatic glucose production, and the thiazolidine diones, or the TZDs, such as rosiglitazone and pioglitazone. All of these medications have sufficiently reduced the risk from 25% to 80% in terms of progression to type 2 diabetes. And most recently, the Diabetes Prevention Program Outcomes Study, or DPPOS, has shown that if individuals can achieve normal glycemic regulation just once over the course of 6 years, that will reduce their risk of progression to type 2 diabetes by up to 56%, which makes it all the more imperative that we are able to identify these patients. One of the barriers to identifying who is at risk of developing diabetes is the use of our traditional clinical tests, which are the 2-hour oral glucose tolerance test, fasting plasma glucose, and glycated hemoglobin A1C. If you were to perform these 3 measurements on the same individuals, they would only agree that an individual was pre-diabetic or at risk of diabetes 8.7% of the time. So being able to identify the right patient to receive this early intervention becomes difficult. It also begs the question, which test is the best for identifying who is pre-diabetic? The reason for this difficulty in identifying the correct patients that are at risk has to do with the underlying biology of insulin resistance. So if we look at a timeline of disease progression and normal, so 30 to 15 years before disease onset, you'll have normal glucose tolerance, where you'll be able to ingest food, glucose is sensed by the pancreas, insulin's produced, glucose is taken up. However, at some point, either your genetics or your environment, which can include increased caloric intake, physical inactivity, obesity, or, as was seen with my mentor, Mike Snyder, a severe viral infection elicits an event that begins a cascade of compensation and adaptation, where insulin resistance will increase and beta cell function will increase through increased production of insulin and increased beta cell mass to compensate for this resistance. In the meantime, glucose levels remain constant as the body compensates, which makes it difficult to detect using our traditional measurements in the clinic. Eventually, the beta cells will begin to fail and give way to increasing glucose levels. During this time, you'll have impaired glucose tolerance, which will eventually give way to uncontrolled glycemic levels as the beta cells fail, where now you will have type 2 diabetes and need therapeutic intervention to bring this glycemic curve back down. So the goal overall is to try to identify when patients are in this adaptation or compensation phase before their beta cells are failed, and intervene in this window of time. So to understand and think about how are we going to detect this, it's important to look at the timeline in the history of diagnostic testing, looking at insulin sensitivity. And it was observed by the Egyptians in 1500 BC that ants were attracted to honey in the urine, as they called it. And this was the excretion of excess glucose that they were observing, or in the beginning, or the first observation of really diabetes. It wasn't until the last century that our standard clinical tests were developed with oral glucose tolerance test in 1923, and then glycated hemoglobin A1C in 1969. This was followed by the development of direct measurements of insulin sensitivity with the insulin suppression test, which was developed here at Stanford by Jerry Reven in 1970. And also the hyperglycemic euglycemic clamp and the intravenous glucose tolerance tests. All of these are clamp assays where patients are treated to prevent glycemic control and are infused with glucose and insulin, and then measuring their ability to take up glucose when giving a steady state. However, these tests are highly invasive and time-consuming and complicated, making them limited in the clinic. So to compensate for this, a number of surrogate measurements trying to use fasting plasma glucose levels, fasting insulin levels, and 2-hour oral glucose tolerance tests have been developed where they're trying to correlate those measurements with these direct measurements. However, most of these surrogate measurements lack correlation and are really measuring hepatic glucose production and not peripheral insulin sensitivity. So the direct measurement we've used is the modified insulin suppression test. And just to give you a little background on how this test works, after an overnight fast, an individual would come in and they're given a continuous intravenous infusion of octreotide, insulin, and glucose over 180 minutes. The blood is then sampled every 30 minutes until 150 minutes, and then every 10 minutes following that. So the administration of octreotide will suppress endogenous insulin secretion so that every individual will have approximately the same steady state plasma insulin level. Therefore, the disposal of glucose will allow us to understand how sensitive an individual is to insulin, where the height of this SSPG concentration is that direct measurement. So the higher the level, the more insulin resistant the person is, whereas the lower the level, the more insulin sensitive this person is. As I said before, despite being a gold standard for quantifying insulin sensitivity, this has limited clinical utility due to the invasiveness, how long it takes, and the complicated nature of having to infuse multiple compounds. So our goal is to develop a new surrogate measurement that we can scale. So our hypothesis was that mitochondrial function is highly sensitive to environmental inputs. And in addition to this, it is deregulated during insulin resistance in type 2 diabetes. So this functional change can manifest in a number of different phenotypes, including mitochondrial DNA mutations, the increased production of reactive oxygen species, the loss of membrane potential during the electron transport chain. It could also be the loss of mitochondrial content due to reduced biogenesis or increased mitophagy, and reduce oxidative phosphorylation or overall loss of metabolic function. So a little more on the mitochondria as a sensor for metabolic function is it is well positioned to integrate multiple inputs from metabolism, starting with glucose and glycolysis, fluxing into the Krebs cycle, and then multiple substrates flowing from the Krebs cycle into the electron transport chain, where oxygen can be measured as a relative measurement of function. It also can be specifically probed, where growth factors such as insulin can be administered to activate glycolysis or mitochondrial function. Glycolysis can also be inhibited given 2-deoxyglucose. And each component or complex of the electron transport chain can also be modified either by administering substrates or specific inhibitors. So the hypothesis we wanted to test first was, what is the impact of insulin on mitochondrial respiration? So to do this, we took a hepatocyte cell line, HepaRG, and we serum starved them overnight, and then used the Agilent Seahorse oximeter to measure oxygen consumption rate. So to begin, we perform 3 basal measurements and then administer insulin. So what you can see is that when given insulin, the green line, there's an acute burst of mitochondrial function, whereas the black line did not receive insulin, stays at the basal level. Next, we administered oligomycin, which inhibits complex 5 and shuts off ATP production. While that is occurring, the rest of the electron transport chain is able to function and continue to put protons across the inner mitochondrial membrane. We're then able to release that potential that's built by administering FCCP, which is a proton uncoupler. And those protons can flood back across, allowing for maximal mitochondrial function or maximal respiration, where we see that cells administered insulin actually have an elevated maximal respiration. And then we're able to turn off the entire electron transport chain by administering rotenone and antimycin A. So together, what we can appreciate is that insulin is able to perform two functions in terms of mitochondrial function. First, it gives a acute increase in respiration, and then allows for a higher maximal respiration and reserve capacity to be achieved compared to unstimulated cells. So together, we've called this assay mitochondrial insulin stimulation test, or MIST. So thinking about using mitochondrial function as a sensor, we wanted to think about the cell type we're using and the dynamic range of detection that we're able to achieve. So here is our data from our hepatocyte cell line HepRG, where the black lines are negative control, no insulin stimulation, green line is our positive control. And the dynamic range between that maximal signal and the stimulated versus the unstimulated is 0.65. We then compared that with skeletal muscle cells, which also were insulin responsive, as you can see by the same type of profile where there's an acute response to insulin and an elevated maximal respiration. However, the dynamic range was smaller, which would be prohibitive for trying to profile and discern the sensitivity across individuals. So because of this, we use the hepatocyte line through all of our future experiments. The next thing we wanted to do was model insulin resistance in culture. So to do this, we used continuous culturing of the cells with insulin. And what you can see is that given the cells continuous exposure of high levels of insulin, like what would be observed in type 2 diabetics or insulin resistant individuals, these cells no longer respond to insulin in the acute and actually have a diminished maximal respiration compared to the starved cells that do not receive insulin. So together, this data told us that our model and assay should be able to detect insulin resistance when given outside perturbations. So to apply MIST to patient samples and to try to test this as a new surrogate measurement of insulin resistance, we have set up our assay similarly, where we starve the cells overnight of insulin, sensitizing them. Then we administered a 1:100 dilution of patient serum to the cells for 4 hours. We then washed out the serum and applied our mitochondrial stimulation test. So all of our measurements were normalized to our positive and negative controls on every assay. So here's our positive no serum treatment with insulin stimulation and no serum treatment without insulin stimulation. So exposure to insulin sensitive patient serum, where their SSPGs are less than 150 mgs per deciliter, you can see the acute response to insulin is there, not as high as the positive control, but still present. And the maximal respiration is still maintained. When you compare that to insulin resistant individuals, serum, when placed on the cells, you lose the acute insulin response, and the maximal respiration is diminished. And then finally, when you compare that with the type 2 diabetic serum treated cells, the acute insulin respiration is lost, as well as the improvement in maximal respiration. So to quantify these on a per individual basis, each dot represents a single individual performed in triplicate, you can see there's quite a span in terms of the acute response amongst insulin sensitive individuals, but significantly more responsive than insulin resistant or type 2 diabetic serum. Similarly, the maximal respiration was even a more drastic phenotype with improvements for the insulin sensitive individuals, and insulin resistant individuals actually had more maximal respiration than type 2 diabetic individuals. So taking these two measurements together, we compared them back to the insulin suppression test. And to our surprise, they were extremely well correlated with an R-squared of 0.7, which is better than any of the surrogate measurements. So suggesting that our assay was capable of being a new functional surrogate measurement of insulin sensitivity. So our next goal was to monitor therapeutic response. And to do this, we wanted to examine type 2 diabetic medication that would improve insulin sensitivity. So looking across the spectrum of potential therapeutics used for type 2 diabetes, they all target different tissues, adipose, muscle, pancreas, but only the thiazolidines or the TZDs, including Resi, clitazone, and pioglitazone, increase insulin sensitivity in glucose uptake in adipose and muscle tissue, whereas all other classes of medications either are cause for more compensation by the body, either through increased insulin secretion or preventing glucose absorption or increasing glucose secretion. So using a cohort of 19 individuals where they had the insulin suppression test performed on them before starting pioglitazone treatment, and then again 3 months after, we performed our MIST. And what we observed is prior to starting pioglitazone, you can see diminished acute response to insulin and a small improvement in maximal respiration. Whereas following post pioglitazone treatment for 3 months, there was an improvement in the maximal respiration. And this change in oxygen consumption rate between individuals was significant. And to give you a feel for the personalized aspect of being able to track people that were responders versus non-responders, here's two examples where on the left, this individual's SSPG started at 164, so they were insulin resistant. And you can see there was not much of a response to insulin, and maximal respiration was diminished. Following 3 months of pioglitazone, their SSPG improved to 57. And now what you can see in terms of their mitochondrial respiration is they're closer to the positive control with their maximal respiration almost completely overlapping. Whereas we can also detect people that do not respond, whereas this individual started at 171, insulin resistant, and was maintained at SSPG at 168. And you can see there was not a lot of movement in terms of their mitochondrial respiration changes with treatment. So to correlate this with the observation in SSPG using the insulin suppression test, both measurements show a significant improvement with pioglitazone treatment, and then the correlation was maintained well. However, using MIST, we observed that there is we we overestimate the amount of sensitivity for people that are sensitive, as people that are with SSPGs around 50, we project them to be even more sensitive than that. And those that are resistant, we tend to especially at the high end where their SSPGs are over 200, we tend to overestimate how resistant they are. So this is an ongoing issue, which might be due to just not enough sampling to get a calibration curve to interpolate from. So the question now that you're probably wondering is, what are we actually measuring in the serum that is able to induce insulin resistance independent of the cellular biology? And what we observed is by doing size fractionation and serial dilutions, that the protein we were looking for had to be highly abundant and greater than 50 kD. So we hypothesized that IgG could potentially be this molecule. So we purified IgG out of our patient's serum and applied it to mist in the same fashion. And what you can observe is the profiles look very similar, where insulin sensitive individuals maintain that acute insulin response and maximal respiration, whereas insulin resistant and type 2 diabetic individuals do not respond to insulin in the acute phase and have not much of an improvement in terms of maximal respiration. We then correlated this with the SSPG measurements performed by the insulin suppression test, and our correlation was maintained at a similar level with an R squared of 0.63. We then wanted to identify what portion of IgG is responsible. So there's the FAB, which is the fragment antigen binding, or the FC. We performed papain cleavage to then purify out 2 FAB molecules per IgG and applied those to mist compared to the full IgG antibody. And what we observed is that the FAB had no effect on the acute insulin response, as the response returned compared to the full antibody, and maximal respiration also was improved compared to the full antibody, suggesting that the FAB portion of IgG was not playing a role in in the induction of insulin resistance. Given that the FC region was involved, we wanted to further validate that. And to do that, we applied FC block, which is a broad blocking agent of FC receptors on the cell surface. So our cells were pre-treated with the FC block, then treated with full serum, and then applied to mist. And what we see is that there is 100% recovery of the acute insulin response for both insulin resistant and type 2 diabetic individuals, as well as the recovery of the maximal respiration, demonstrating that the FC region and FC receptors are required for the induction of insulin resistance. So what could be different about the FC region of IgG? And glycosylation has been linked to many effector functions of the FC region. So in order to profile this difference, we performed lectin ELISAs across our insulin sensitive and insulin resistant individuals, looking at terminal sialic acid as well as terminal galactose. And what we observed is there's an enrichment of insulin within the insulin sensitive individuals of the presence of either terminal sialic acid or galactose, which then made us ask whether insulin sensitivity or sialic acid as an anti-inflammatory molecule or galactose are sufficient to elicit insulin resistance. So to test this, we employed IgG B cell null mouse. So we used the mu-MT and then purified mouse IgG. And the IgG was either left as a wild type pool of glycoforms or was treated with neuraminidase to remove all terminal sialic acids. These mice were then administered either the wild type or the acylated neuraminidase treated IgG in the fasted state. And we measured glucose levels immediately following administration. And what we observe is the acylated IgG induced a rapid increase in glucose levels immediately following administration compared to the wild type pool of glycoforms. We then went back and tested the glucose levels 3 hours after injection, and they remained significantly elevated compared to the wild type pool of glycoforms. We also looked at insulin levels, which were also elevated, suggesting that these mice, when administered the acylated IgG, was inducing an acute phase of insulin resistance. Following this, we then went back 24 hours later and performed glucose tolerance tests. And what we observed is that the mice receiving the wild type glycoform pool of IgG were significantly more glucose tolerant than those receiving the acylated IgG. So together, these results demonstrate that acylation of IgG is sufficient to induce insulin resistance in vivo. So our model from this data is that when you are insulin sensitive, you have a more anti-inflammatory pool of IgG glycoforms where there's the presence of sialic acid. And as you progress to insulin resistance and type 2 diabetes, you're promoting a more pro-inflammatory state, which results in the progressive trimming of glycans to a more pro-inflammatory molecule that then has repercussions on insulin sensitivity. So I hope you can take away from this seminar that mitochondrial function is sensitive to changes in insulin sensitivity, that the mitochondrial function can be used as a functional surrogate of insulin sensitivity and is transferable through serum, and that MIST is capable of monitoring changes in an individual's insulin sensitivity in either at baseline or through therapeutic response, and that IgG glycosylation states are correlated with insulin sensitivity and are sufficient to alter glycemic regulation in vivo. And with that, I'd like to thank members, my mentor, Mike Snyder, and Neil, who helped develop the MIST assay, my collaborators, Josh Knowles and Fahim Abbasi, who provided samples, as well as Tracy McLaughlin. And then the in vivo studies in the MUMT mice were performed in collaboration with the ANS lab and Jenny. And then funding, NIH, Spark, and then PHIND. And with that, I'll take whatever for questions.
All right, everyone. Yeah, so please either enter your questions in the Q&A or feel free to raise your hand and we can call on you directly as well. Any questions for Andrew?
Great job, Andrew. This is Josh.
Oh, hi.
Sorry, I'm in clinics. I've been muted, but I was following it. I think it's fantastic, as I've told you before.
Mm-hmm.
Talk to us a little bit about your plans for potentially, scaling this so that we can assess insulin sensitivity. Getting back to the idea of having a non-invasive measure of insulin sensitivity that outperforms all the other surrogates that we have.
Yeah. So I think definitely a more detailed profiling of the glycoforms is necessary. There's some data on if you have bisecting glycosylation, that will oppose the sialation. So there's definitely more glycan and glycoforms that could be profiled to increase the power, which would require doing mass spec. So.
Let's see.
Do you think that it's impossible to scale the cellular assay? Do you think you have to profile the glycosylation states?
I think the glycosylation states is probably the easier path forward, mainly because these cells grow really slow. And I think that's part of why they're able to maintain their responsiveness to insulin and serum, because we've tried a number of other cell lines and we do not see this same phenotype.
Excellent. Okay, now we have I think we've inspired some questions with that discussion, so thanks so much for that. Do you think restoring sialylated IgG will work as a therapeutic in humans or removing asialylated IgG?
Yes, I mean, I have data not presented or not here, but where if you apply the IVIG paradigm where you administer a high dose of IgG, we can resensitize DB mice, and that provides a long-lasting therapeutic response. And we've shown that those mice also have systemic loss of sialation on their IgGs.
And I guess a related question is, how much sialation occurs within B cells versus extracellularly?
That's a great question. And it's a hot topic right now that it's believed that when the B cells being made, the IgG is being made, it's being secreted and being glycosylated. But there's been a number of papers in the last 2 years showing that the sialotransferase ST6-Gal1 is actually secreted and is in circulation and is capable of sialating. So it's unclear how much sialation is happening extracellularly.
Excellent. And then there's also it says plasmapheresis in humans, question mark.
Maybe, I think IVIG is probably easier.
Sure. And then for your in vitro assay, what type of hepatocytes do you use? Healthy or diseased or mortalized cell line?
So they're a healthy, healthy, short-lived immortalized cell line. They're HEPA RGs. They're mainly used for clinical drug development because they maintain most of their metabolism.
Okay, great. And then have you studied the effect of acute infection, which increases IgG on insulin resistance and serum glucose levels?
Yes, I've done some preliminary studies in mice, and we've shown that they do become insulin resistant. IgG loses its silation as well at the same time. So, yes.
Let's see. And they said, I'm wondering if you tried missed assay in hepatocytes from diabetic or aged hosts who might have a defective mitochondria.
We have not done that.
Let's see. Let's see. So the IgG overwhelms the effect of exogenous insulin. The serum levels of insulin must have been somewhat different in IR versus IS.
For the assay, I mean, the dilution is so low. And also we've done real-time monitoring where we've injected the serum onto the cells and measured, and there is no response. So yeah, the insulin effect in the serum is minimal.
Okay. It says, so for patients with IgG overproduction, like in multiple myeloma, do they have more insulin resistance?
I don't know. That's a great question. I haven't seen anything suggesting or looking at that, I think. So that's something to look into.
Excellent. Any other questions for Andrew? Okay, well, thanks so much, Andrew. I mean, this was a really excellent mechanistic talk on, your journey and discovering, looking at this biomarkers and future directions. So very impressive work. We're really excited to have you share it with our forum.
Thank you. Thanks for having me.
All right, thanks everyone. Have a great day.